Hi, see you again with me, Andrew, on the Bright Side of Life channel.
There's always a bright side behind the darkness. So let's start from here.
This time we will discuss an information that a lot of people are looking for. What's that? Yes, information about tumor immunology. Before we enter the discussion, don't forget to subscribe so you don't miss important information about various facts of life around us. The immune system's other key function is to detect and eradicate tumors. Immune surveillance is the term for this. Tumor cells that have been transformed express antigens that are not seen in normal cells. These antigens appear strange to the immune system, causing immune cells to target the altered tumor cells. Tumor antigens come from a variety of places. Some are derived from oncogenic viruses like the human papillomavirus, which causes cancers of the cervix, vulva, vagina, penis, anus, mouth, and throat. And others are the organism's own proteins, which are present in low amounts in normal cells, but are abundant in tumor cells. One example is an enzyme called tyrosinase that, when expressed at high levels, transforms certain skin cells, for example, melanocytes, into tumors called melanomas. A third possible source of tumor antigens are proteins normally important for regulating cell growth and survival, that commonly mutate into cancer-inducing molecules called oncogenes. The main response of the immune system to tumors is to destroy the abnormal cells using killer T cells, sometimes with the assistance of helper T cells. Tumor antigens are presented on MHC class I molecules in a similar way to viral antigens. This allows killer T cells to recognize the tumor cell as abnormal. NK cells also kill tumorous cells in a similar way, especially if the tumor cells have fewer MHC class I molecules on their surface than normal. This is a common phenomenon with tumors. Sometimes antibodies are generated against tumor cells, allowing for their destruction by the complement system. Some tumors manage to avoid the immune system and develop into cancers. Tumor cells frequently have less MHC class I molecules on their surface, allowing them to slip past killer T lymphocytes. Some tumor cells also produce chemicals that decrease the immune response, such as the cytokine TGF, which reduces macrophage and lymphocyte function. In addition, immunological resistance to tumor antigens may develop, allowing the immune system to stop attacking tumor cells. Paradoxically, macrophages can promote tumor growth when tumor cells send out cytokines that attract macrophages, which then generate cytokines. And growth factors such as tumor necrosis factor alpha that nurture tumor development or promote stem cell-like plasticity. In addition, a combination of hypoxia in the tumor and a cytokine produced by macrophages induces tumor cells to decrease production of a protein that blocks metastasis and thereby assists spread of cancer cells. Anti-tumor M1 macrophages are recruited in early phases to tumor development but are progressively differentiated to M2 with pro-tumor effect, an immunosuppressor switch. The hypoxia reduces the cytokine production for the anti-tumor response, and progressively macrophages acquire pro-tumor M2 functions driven by the tumor microenvironment, including IL-4 and IL-10. Cancer immunotherapy covers the medical ways to stimulate the immune system to attack cancer tumors. Are you still confused? I hope what I explain can make you understand. But okay. We'll try to explain in a slightly simpler form. Let's continue a little more. Cancer immunology has tremendous therapeutic promise, as the immune system has the potential to find and completely eliminate tumor cells throughout the body, and provide long-lasting immunity from cancer. Researcher currently investigating how immune cells and tumor cells interact during tumor development and how productive and non-productive anti-tumor immune responses affect the development of cancer in mice. Natural roadblocks that inhibit the immune system from attacking the body stand in the way of powerful anti-tumor reactions by immune cells. Complete removal of these barriers could be fatal. Understanding how to relax immune system controls and allow anti-tumor responses to occur is crucial to successful therapeutic stimulation of anti-tumor responses. The Cancer Immunology group focuses on two main groups of cells, T cells and NK cells. The interactions between tumor cells and T cells have been studied for many years. However, the reason why T cells do not naturally eliminate tumor cells as they do infected cells remains unclear. Researchers also focused on natural killer NK cells, thought to kill tumor cells in the body, and finding ways to target these cells towards tumors. As T cells and NK cells play an important role in lung cancer development, and modulating these pathways may be a viable therapeutic strategy for improving outcomes for patients with lung adenocarcinoma. The group is also interested in learning more about the involvement of other innate immune cell types, including macrophages and dendritic cells, in recognizing tumor lesions and instructing adaptive anti-tumor response. It's a good idea you also watch my video about white blood cells. I hope that watching it can add to your insight, which has little to do with the discussion in this video. In a healthy adult, white blood cells make up about 1% of total blood volume, which is significantly less than red blood cells, which make up 40% to 45% of total blood volume. This 1% of blood, however, has a significant impact on health because immunity is dependent on it. Leukocytosis is defined as an increase in the number of leukocytes above the maximum limit. When it occurs as part of a healthy immunological response, which occurs frequently, it is considered normal. When it is malignant or autoimmune in nature, it can be aberrant. Leukopenia is defined as a drop in white blood cell count below the lower limit. This is a sign of a compromised immune system. The physical appearance of a blood sample after centrifugation gives rise to the term white blood cell. The buffy coat, a thin, generally white layer of nucleated cells between the sedimented red blood cells and the blood plasma, contains white cells. The scientific word for this type of cell is leukocyte. It comes from the Greek origins leuk, which means white, and cyte. I hope it's useful, and don't forget to subscribe. See you in my next video.